ना कि छोटे से कीड़े को मारने के लिए बुलडोजर चला दिया. So like that, you are delivering 500 milligram 4 times a day or 3 times a day for killing that amoeba, right? Because why? Question is why 500 milligram of the drug is required to deliver through the oral route, to be administered through the oral route? Because for delivering effective concentration of the metronidazole that is required in the nano quantity, right? You will require a large, because most of the drug which you most of the metronidazole which you deliver through the conventional dosage form via the oral route, that it will be absorbed from the upper part of the GIT. When I say upper part of the GIT, it means stomach or small intestine. Most of the administered metronidazole is absorbed from the stomach, either from stomach or small intestine, and it will reach to the systematic circulation, and from systematic circulation it will reach to the colon, right? But if same metronidazole in a very less quantity, microgram concentration, if you deliver it through the colon specific delivery system, no drug will be released in the upper part of the GIT. Means no drug will be released in the stomach and small intestine. The whole drug will be released in the colon, which is the major site of the protozoal infection, right? So the whole drug is we deliver. So in a small quantity, you can rectify this problem, right? So this is on less amount of the drug will be reaching to systematic circulation. So you will have, if it is delivered through the colon specific drug delivery system, you will have less side effect as compared to the conventional dosage form, right? What's supposed to be the drug candidates for colon delivery? Those drugs which are having poor bioavailability from the stomach and small intestine, like peptides, that can be delivered through the colon, or those drugs which are having deleterious effect on gastric mucosa, that can also be delivered through the colon, or drugs which are required for the treatment of diseases associated with the colon, like amoebiasis, colon cancer, ulcerative colitis, inflammatory bowel disease. For the treatment of these disease, you will require to deliver drug to the colon, right? There are some examples of the drug candidates which are used for colon-specific drug delivery system. Some digestive enzyme supplementary, sulfasalazine, that is the epsilagine, valsalagine, mesalagine. These are the 5-aminosalicylic acid for drug. Prednisolone, that is steroid. These are used for the treatment of irritable bowel disease, for topical action. 5-fluorouracil for the treatment of colorectal cancer, for systematic absorption. Non-steroidal anti-inflammatory drugs preventing gastric irritation, typhoid, for the delivery of the vaccine, oral delivery of the vaccine. Insulin, oral delivery of peptides. So these are the candidates can be used for designing the colon specific delivery. They can be delivered through the colon. So colon specific drug delivery system, if it is administered through the oral route, they are having, they are releasing exclusively in the colon, right? Drug will be exclusively released in the colon. It will not be released in the upper part of the GIT. And so the colonic bioavailability will increase, enhance therapeutic, it will enhance the therapeutic efficacy by localizing to the colon, reducing systematic adverse effect by reducing the absorption of the drug from the upper intestine, maximize drug utilization, reducing dose of the drug, maximizing drug retention time in the colon, right? So these are the advantages if we develop colon-specific drug delivery system. Now, question is, how will you develop colon-specific drug delivery system? There are some emerging technology, apart from just taking a tablet, coating it with some anti-coated polymers. There are some emerging technology apart from that for colon targeting of the drug. That is a prodrug approach. Site-dependent drug delivery system. Product approach means azobond product, glycoside product, cyclodextrin product, dextran product. Size-dependent drug delivery system by using the nano drug delivery system of suitable polymers. Surface charge dependent drug delivery system because colonic membrane is having both positive and negative charge. So both type of the delivery system beneficial for targeting drug to the colon. pH dependent drug delivery system, those polymer which are dissolve above 6.8 pH, then that will be good for the delivering drug to the colon. Biodegradable drug delivery system. So some polymers, polysaccharides like pectin, guargum, chitosan, chondroitin sulfate, they can be used for fabricating drug delivery system because for digesting these polysaccharides, the polysaccharide enzyme is present or released by the colonic bacteria only. So we can trigger drug release in the colon. Redox drug delivery system, this is especially for the case of the inflammatory bowel disease where reactive oxygen species level is increased. So that ROS can be used for taking drug release from the redox drug delivery system. Active targeting, that is especially meant on which I received the President Award, that is the active targeting dependent drug delivery system for the treatment of the colorectal cancer. Using some ligands, we achieved the effective targeting of the colorectal cancer. By designing nanocomposites, some using some earth matters that are beneficial for the treatment of inflammatory bowel disease, for reducing the inflammations. There are some earth matters which are reducing the inflammation that can also be used. So along with some polymers, we can prepare nanocomposites. Dendimers, in the skeleton of dendimers, if we have azo bonds, then that dendimers we can prepare for the colon delivery and time release system. So I think Ganesh, can you tell me how much time left?
Sir, if possible, please complete within 15 minutes, sir.
Okay. So, well, by 15 minutes, it will be over, right? So there are different approaches. I will take only one example of each technology. So this is the example of the prodrug, azo bond prodrug of 5-aminosalicylic acid. This is the sulfasalazine. It is having azo bond. This is the N double bond, and this is the azo bond. And sulfasalazine, this drug is because of presence of the azo bond, it is not absorbed in the upper part of the GIT. When it reaches to the colon, the azoreductase enzyme will be released by the bacteria present in the colon, and that azoreductase enzyme cleave this bond, right? So that azo bond will be cleaved, and you will get one molecule of 5-aminosalicylic acid and one molecule of the sulfapyridine. The side effect of this sulfasalazine, most of the side effect is because of the formation of sulfapyridine, right? So these side effects are overcome by making the another, means sulfapyridine is replaced by some amino acid, 4-aminobenzoyl beta-alanine or 4-aminobenzoyl hippurate, or by making prodrug by taking the 2 molecule of 5-aminosalicylic acid. That is in case of the osalazine, right? It is a dimer having 2 molecules of 5-aminosalicylic acid, and these 2 molecules of 5-aminosalicylic are joined by the azo bond. And azoreductase enzyme cleave this, you will get 2 molecules of the 5-aminosalicylic acid. Then glycoside prodrug, as I told you, glycosidase enzyme is released by the microorganisms present in the colon. So this is the example of dexamethasone 21D glucoside, and this is cleaved. This is cleaved by the glycosidase enzyme. You will get the parent drug, that is the dexamethasone in the colon, right? Then cyclodextrin is the beta cyclodextrin is generally used for enhancing the water solubility. And here, if it is encased inside this beta cyclodextrin molecule, right? If the drug is encased inside this molecule, beta cyclodextrin molecule, the solubility will enhance. But if it is conjugated with the OH group present over the surface of this beta cyclodextrin molecule, so that conjugate will become a colon-specific drug prodrug. Means this we cleave this a group of the beta cyclodextrin substitute with the diphenylyl acetic acid through an ester. So this amide linkage is cleaved by the cyclodextrin enzyme released in the colon, right? So and cyclodextrin is excreted unchanged with the feces. Similarly, dextran product is prepared then by succinate and glucarate bonding, and we can make it colon specific. And size dependent means if you reduce the size, then naturally the residence time of the drug delivery system will improve, and the EPR effect is also enhanced, right? And it allows preferential uptake of nano-sized particle by immune cells at the inflammation. This is generally in case of the inflammatory bowel disease, and avoid rapid carrier elimination by the diarrhea. In case of the inflammatory bowel disease, there is a rapid diarrhea. So we can avoid the clearance of these nano delivery system, elimination of nano delivery system from the colon, right? And so this is size dependent nano delivery system. By reducing the size, we can enhance the resident times of the drug, especially in case of the inflammatory bowel disease. Surface charge in nanocarriers, there are two types of the carriers, positive charge or negatively charged nanocarriers can be used. There is an enhanced adherence to the inflamed mucosa of the colon, especially in case of the positively charged nanocarriers. But in case of the inflammation, the certain proteins are overexpressed, like eosinophil, casein proteins, and transferrin. So negatively charged nanocarriers also adhere by these proteins. So if you see the adherence level of the different charged particles, nanoparticles, the negatively charged particles are more adherent as compared to the positively charged particles. Then pH-dependent drug delivery system we can develop. pH-dependent drug delivery system means just using the pH-sensitive polymers, those polymers which are dissolved at the pH 6.5 or above. Then we can deliver drug in effective concentration to the colon, right? Then biodegradable drug delivery systems, some azoaromatic polymers or some polysaccharides that can be used for fabricating the nano drug delivery system or colon-specific drug delivery system. Say azoaromatic polymer, they are having the azo bond, number of azo bond in the skeleton of azoaromatic polymers. And if we fabricate any drug delivery system using this polymer, then this polymer will not release drug in the upper part of the GIT, but when it will reach into the colon, azoreductase enzyme will be acting on this azoaromatic polymer, right? It will cleave the azo bond, so you will have a porous skeleton and drug will be released. Similarly, in case of the alginate, pectin, and dextran, these are the polysaccharides. And polysaccharide enzymes will degrade these alginate, pectin, and dextran and release the drug. Say, using the azoaromatic polymer, we can fabricate nanoparticles, right? And fill in the capsule, and capsule will be entry-coated, so it will be intact in the small intestine, in stomach and small intestine. But when it will reach in the distal part of the small intestine, intercoating will dissolve, and capsule, uncoated capsule will reach into the colon, right? Capsule cell will be dissolved in the colon, and the nanoparticles of azoaromatic polymer will come out, and azoreductase enzyme will be acting and degrade the azoaromatic polymer, and that will be released, right? We can embed the drug in the polysaccharide matrix also, and we can prepare a tablet or microcapsules, matrix type of microcapsule, micro spheres, and embed the entrap the drug, and that will be intact in the stomach and small intestine. But when it will reach into the colon, the polysaccharide enzyme will be acting, and the polysaccharide matrix will be degraded and drug will be released. So this is, I'll discuss only the one, our research that we prepared calcium pectin in microspheres containing the anti-cancer drug. So here the pectin solution we prepare, 3% pectin solution we prepare in phosphate buffer saline, and we added 280, 1%, and drug was dispersed, mixed well. And then this dispersion is dispersed in the isooctane having span 80, right? Stir at 1500 RPM for 10 minutes. We'll get the emulsion, say water and oil emulsion, right? Again, further stir for 10 minutes, right? And this in this dispersion, we add 35% calcium chloride solution and stir it continuously for 10 minutes. Because of presence of the calcium ion, the pectin will be reacting, and this calcium pectinate microcell will be precipitated. So you will have solid microspheres, and will centrifuge it, wash it properly, and these are the same photograph of the calcium pectinate microspheres having. This is without drug, and this is lower one is with drug. Right. Then just to ascertain that whether drug is released in the upper part of GIT or not, right? Where drug will release, in which part of the drug is released. So these in vitro drug release studies were performed in different simulated GIT fluid, say simulated gastric fluid, pH 1.2, right, at for 2 hours. Then simulated intestinal fluid, simulated gastric intestinal fluid in a mixture of the simulated gastric intestinal fluid, pH 4.5 for 3 to 4 hours. Then at pH 7.5, 5 to 8 hours, and simulated colonic fluid that is having pH 7, 8 to 10 hours. And we find that after the 8 hours, sufficient amount of that drug will start releasing, right? And then we also perform in vivo studies by at different time interval. We sacrifice the animal and isolated the GIT and isolate different parts and see the drug content in the different part of the GIT. Then after the 8 hours, we find in the colon large amount of the drug is present. Similarly, guar gum microspheres we can prepare by using the suitable techniques. This is the emulsion solidification technique using the chloraldehyde as a reidizing agent, cross-linking agent for solidifying the guar gum. So we prepare guar gum, and then these are the guar gum microspheres with drug, without drug, and with drug. And again, just to see that these guar gum microspheres, whether releasing drug in the colon or not, and if it is releasing the colon, how much drug is released, and what will be the effect of the cecal content, microbial content, or enzyme content. So we perform in vitro release studies in simulated colonic fluid with or without cecal content, and we see that in the presence of cecal content, the drug release is increased. Then we induce enzymes. We also induce enzymes, that is the polysaccharide enzyme in the guar gum by just feeding the animals 4 or 5 days the guar gum. So the by feeding the guar gum to the animals, the the concentration of polysaccharide enzyme is increased in the colon, because for digestion of the guargum, the bacteria will release more amount of the polysaccharide enzyme. So the concentration of polysaccharide enzyme will increase. Then we sacrifice the animal, isolate the cecal content, and mix it with the simulatory colonic fluid, right, and perform in vitro release strategy. Then we'll find that as on increasing the concentration of the cecal content from 2 to 4, that drug release in 21%. So that is due to the increasing the polysaccharide enzyme. Say redox, as I told you, in case of the ulcerative colitis, 10 to 100-fold ROS content is increased. So if you use some redox drug delivery system, like a delivery system is made up of a polymer that is known as thioketal. Thioketal is oxygen sensitive polymer. So in presence of ROS, this thioketal is degraded and release the drug. This is active targeting. How this, yes. This is the liposomes based colorectal cancer targeting. So we prepare liposomes and incorporate drug, anti-cancer drug, and surface of the liposomes is encoded with the folic acid. And these liposomes are entrapped in the calcium alginate beads, right? And this calcium alginate beads are further coated with the anti-coated polymer like utrazit S, right? When this these beads deliver through the oral route, right, it will be intact because it is, these beads are anti-coated, so it will be intact in the stomach, right? But when it will reach into the ileocecal region where pH is greater than 6.5, the anti-coating will dissolve, right? And the calcium alginate, by the time calcium coating is dissolved, the calcium alginate will reach into the colon. In the colon, polysaccharide enzyme will be digesting the calcium alginate, right? And release the surface modified liposomes. And since the surface of the liposomes is modified by the folic acid, so the receptor of folic acid are overexpressed on the colonic tumor, right? So it will be taken up by the colonic tumor, and so that there is a effective or dual drug delivery of the dual targeting, not dual drug delivery, it's a dual targeting of the drug. Say first is targeting to the colon, and then in colon it is further targeted to the colorectal cancer. So effective targeting can be achieved by using this technology. Multi-particular system we can use means apart from using these polysaccharides enzyme, polysaccharides, we can use pH-sensitive polymers. So double protection is given so that no drug will be released in a part of the GIT, right? So in this case, we prepared Chitosan microspheres, and these are the same photograph of the Chitosan microsphere. Then again, just we perform different release studies just to ascertain that what will be the effect of the cecal content, how much drug will release, right, in what time. So the different studies, in vitro release studies are performed. This is the very new nanocomposite. One of my PhD student prepared this nanocomposite. So we filed the patent also. Here we prepared a layer nanocomposites. Chitosan has been intercalated by ionic exchange by negative charge montmorillonite, right? So Chitosan montmorillonite nanocomposites remarkably reduce the ulcer index 3.8 as compared to the Chitosan nanoparticles that is having this 3.03 and mesallagine, which is having ulcer index 9.85. So we can see that It is a 3 times effective as compared to the mesology. Right? So what will be the use of using this montmorillonite? Montmorillonite shows anti-inflammatory activity by adsorbing luminar antigen, increase of the colonic mucin level, and possibly a direct modulating effect of cytokine production by mucosal cell. It is well documented that epithelial barrier defect induced by the pro-inflammatory cytokines has been restored by the montmorillonite. So using the montmorillonite, not only preparing the nanocomposite, it is also used as a drug for reducing the inflammation. Dendimers, in the skeleton of the dendimers, if you prepare dendimers, and in the skeleton of dendimers, if azobond are, because in the dendrimer there are a number of amino groups. So if we prepare, if we put azobond in the skeleton of the dendimers, we can make dendimers colon specific one. Some time release system just to delay the release of the drug, so that by the time your delivery system will reach to the colon, say the whole transit time is say 6 to 8 hours. If you are able to delay the release 6 to 8 hours from the drug delivery system, we can achieve drug delivery, effective drug delivery to the colon. Say that is the pulsine cap. It is having the capsule body, which is having the water insoluble body, right? And the mouth is capped with the hydrogel, right? And then it is capped with, and it is intercoated. When it reaches to the ileocecal region, intercoating is dissolved, cap is also dissolved, and this hydrogel will expose to the water, right, in the ileocecal region, right, and will start expanding, right? Due to the swelling, it will start expanding. So there is a slow swelling of hydrogel leads to a lag time that is predetermined by length of the hydrogel. So that lag time we can monitor by changing the length of this hydrogel. So lag time we can monitor. So by swelling this, slowly swelling, the length is increasing, and by the time this whole system will reach into the colon, right? Then by hydrogel plug, on further swelling, the hydrogel plug is ejected and drug will be released. And by that time, your system will reach into the colon. So thank you very much. In the last, I would like to say, let us not follow a path set by the others. Let us set a path for others to follow. Thank you very much.
So thank you very much, sir. And I know that the lecture was very, very excellent, and Professor Nk Jain is a very learned person and scientist. Obviously, he is the master of the colon drug delivery system. At the time I was the student, so I know that the very, very interesting topic, and she did a lot of job work on this topic, and also recipient of the award. So no doubt, Professor Genser, he is the update the each and everything, because I know that time is not allowed to me to go to one by one slide and justify each and everything. So I just request to the audience, if you have any question, so because time is very short, so I will take only the one question. So any question in the chat box, or any person want to direct ask with the expert?
Say, okay, there is a question, say IUC Aurora.
Yeah.
Right? And she asked about the, is there any difference between nanoparticles and nanocomposite? Yes, there is a difference between nanoparticles and nanocomposites. Nanocomposite is just the mixture of the material, right? And nanoparticles, they are nano in size. They are having a matrix system, right? They are having a matrix system. So these will be the nanoparticles. We call if they are in nano, both are the nano in size. Only the method of preparation is different. Nanoparticles and nano composites, right? So nano composites, we can say it's a mixture of the some materials. It is having some the mixtures are bound together by say some electric charges, right? But in nanoparticles, these nanoparticles are prepared by some material, right, of a single material. So that will be, it may be mixture of the materials also, but they are the physical mixture. And here nanocomposites are they joined together by some charge interactions.
So thank you very much, sir. Very nice lecture, and Professor Genser included each and everything, prodrug approach, anatomy, physiology of the colon, as well as the physics, and from basic to the up to the mark. So thank you very much, sir. And this lecture and this talk, obviously it will be the very, very fruitful to the all audience, researcher, and you have also given one of the approach, new approach, so that number of the students, they will be benefited to this. Over to the organizer.
Thank you very much, sir. Thank you very much for your informative session. Now, I would like to request our respected principal, sir, to felicitate the speaker of the day, Dr. Sanjay Kumar Genser. Sir, please, over to you.
Thank you, Dr. Rekha. And thank you very much, respected Sanjay Genser. As you all are aware that Dr. Sanjay Genser is my teacher, my guru. So it's on behalf of entire IIP, I would like to felicitate Dr. Sanjay Genser. Sir, please accept a token of love. I'm felicitating in form of e-certificate. Dr. Rekha, please share. Thank you very much, sir, once again.
Thank you very much, Dinesh,
Sir,
for giving me opportunity to deliver or share my experience with the audience. And I'm just on the screen. I was looking the Pradeem Mishra is there.
Yes, sir. He is the next speaker.
Acha, okay. Very good. So Pradeem Mishra, just convey my regard to Pradeem Mishra also. Right? Namaskar, Mishra ji.
Namaskar, sir. Namaskar.
How are you?
I'm doing good.
Good.
And,
so you are still in Bhopal?
Yeah, I'm at Bhopal. Yes.
Good. So well, thank you, Dinesh.
Thank you, sir. Thank you, sir.
Thank you very much.
I would also like to thanks Professor Dr. G.D. Gupta, Director ISF Moga, for sparing his valuable time and chairing the session. So please accept, sir, the certificate of appreciation. So thank you very much for coming and giving your valuable time. Thank you, sir.
Thank you very much. Thank you. Thanks to all.
Over to you, Dr. Rekha. Now you can continue with the session.
Thank you very much, sir.
Dinesh, I take your permission to leave this webinar because I have a meeting at 1 o'clock.
Sir, thank you, sir. Thank you.
Pradeem.
Sir.
नमस्कार सुमित नमस्कार आई एम फाइन आप कैसे हैं?
मैं ठीक हूँ सर.
गुड. सागर आना नहीं हो रहा है
सर? आऊंगा. I will come on Diwali ना, so I will meet you definitely.
Sure. Okay.
डॉक्टर जी नमस्कार.
Thank you very much. नमस्कार डॉक्टर साहब. नमस्कार. हमारे तो इतने पास हो गए सर, बहुत कम दर्शन देते हैं.
नहीं.
तुम्हारी तो आदत है पकड़ के ला सकते हो.
जी.
सागर से तुम पकड़ के ले जा सकते हो वहां पर, तो फिर बहुत पास में है.
अमृतसर 100 किलोमीटर ही है.
हाँ. Okay, thank you very much.
Okay, thank you very much, sir.
Thanks all. God bless you.
Thank you very much. Now, moving forward, I would extend my heartiest welcome to our second chairperson, Dr. Shubhit Kumar Genser. And it's my immense pleasure to introduce Sir to the audience. Sir is professor at Department of Pharmaceutical Sciences at Guru Nanak Dev University, Amritsar. Sir is currently director at Directorate of Open and Distant Learning and also at Directorate of Online Studies. Sir is presently member of Secretary and in charge of Central Animal House Facility, Guru Nanak Dev University, Amritsar. He is also in charge of BSL 3 facility, Guru Nanak Dev University, Amritsar. Sir is having 20 years of teaching and 18 years of research experience. He has published many research publications in journals of national and international repute. Sir is also a member of editorial board of many journals and is reviewer for many international journals like Elsewhere, Taylor and Francis, Springer, and Bantam Science. Now, I would request Dr. Shubhit Kumar Genser to kindly introduce our eminent speaker, Dr. Pradeem Kumar Mishra. Sir, over to you.
Thank you, Dr. Rekha, for such a nice word in introduction. First, I'm highly thankful for Dr. Dinesh Mishra and his team and the whole management of the IIP to invite me as a chairperson for this particular conference. The theme of the conference, Translational Research for Nanomedicine, is almost similar in the area we are working here. And I want to share with all of you that just to promote this translational research, I have set up here the one center that is called Center for Translational Research in Health Science under the RUSA 2 scheme, under which we got around 10 crore rupees just to promote this particular types of the things. Because what I feel that this particular translational word is very, very important to further collaborate with the industry. So I feel privileged and honored to introduce today's next speaker, Dr. P.R. Mishra, before the audience. Sir is currently serving as Scientist F and Head, Department of Molecular Biology at ICMR National Institutes for Research in Environmental Health, Bhopal. Sir is a Fellow of National Academy of Medical Science and a recipient of Fulbright Nehru Fellowships for his seminal contribution in the field of environmental epigenetics and translational research, especially in the area of the nanoscience. He has awarded African Indian Mobility Fellowships Award from DVD. He is also a recipient of many of the awards from the prestigious academic and research institutions and academy in India as well as abroad. Recently, his research group invented a novel role of mitochondrial induced epigenetic modification in airborne particulate matter exposure and development of low-cost nanobiosensor for predicting future disease susceptibility using cell-free circulating mitochondrial epigenetic signatures as biometrics. So this is one of the best example of the translational nature of your research. He has received more than 12 major research projects being funded by the national agencies such as ICMR, DHR, DST, DBT, and DAE, and international bilateral projects including Indo-EU and Indo-US and Indo-African. Overall, Dr. Mishra has authored more than 150 peer-reviewed research publications and NCBI PubMed Index Journal of international repute. His current H index is 39. That's I really congratulate you to getting this H index to up to this level. I10 index is near 100 and total citations around 4,000 with a total cumulative impact factor of 425. Sir has 4 patent publications and co-edited 2 books Sir has supervised 11 students for PhD degree, and more than 100 students have worked under his mentorships for their postdoctoral fellowship, internships, and the postgraduate dissertation. Sir, your contributions in the field of the nanomedicines and especially in the epigenetics means it's a significant contribution in this field. So I'm inviting you on the behalf of IIT to share your views, especially in this translational nature of the research, especially special emphasis on the nanomedicine. Thank you very much, sir. Now I'm inviting Dr. Mishra for sharing his view and his presentation. Thank you very much.
Thank you, Dr. Subit, for a wonderful introduction.
Thank you, sir.
Let me begin with a congratulatory note to Dr. Dinesh Kumar Mishra and Dr. Pankaj Dikhet and all the associate members of all IIP Indore. I guess they have done a wonderful job by organizing this webinar in a fabulous manner during this times of unprecedented uncertainties of COVID-19. The fourth wave is looming. It's coming very fast. So I'm not sure whether it would have a detrimental effect as we saw in the third wave. But yes, ICMR is all prepared to take up this challenge. So with the thanking note to Dr. Subir, Dr. Dinesh, Dr. Pankaj Dikhet, and Sri Arun Bhatnagarji, I would like to present my views on low-cost innovations in predicting the future disease susceptibility with the help of few nanobiosensors, examples of few nanobiosensors. I'm currently working as scientist F and head in the Division of Environmental Biotechnology, Genetics, and Molecular Biology at ICMR near Bhopal. Well, we are uniquely positioned, isn't it? In Bhopal. Bhopal is Madhya Pradesh is the heart of India. So Bhopal is again the center of India. So Hindustan ke dil mein hum base hue hain. So this institute was commissioned in 2010 with a broader mandate of researching the environmental for healthier lives. We have 6 constituent laboratories like environmental biochemistry, environmental molecular biology, biotechnology, toxicology and pharmacology, and there are several other areas in which the institute works. So the importance of environment on human health has been realized very lately, because with the climate change and air pollution, news headlines almost hitting our TV screens almost on a daily basis. You're looking every day on newspapers and TV sets about and hearing about climate change, hearing about air pollution, particulate matter. So this has, kind of immensely catalyzed our thinking towards the detrimental effects of environmental pollutants on human health. So there is not an out of doubt that what makes us who we are might be starting much before our birth, and it might not all be related to our genes alone, because genes and environment together might be playing an important role to shape up the human health and disease index. So Professor Dinesh has allotted me around, how much time do you have? Around 30 minutes, 40 minutes? I have 30?
30 minutes, yes, sir. Yes, sir.
Okay. So I have kind of designed my talk in which, first of all, I would like to highlight few generalized aspects on environment, its susceptibility windows, and how to basically address the development of the disease phenotype. It is extremely important because chronic diseases, the non-communicable diseases, they have an immense burden on the health indices of our country. I'm also going to throw a small light on how does mitoepigenetics play a role in all these aspects. whether we can use cell-free circulating mitoepigenetic signatures as biomarkers for disease screening. And lastly, I would try to highlight few of our things in which we have developed nanobiosensors taking help of the analytical frameworks, and that's the kind of translational research perspective that I'm going to present before you. In relation to this presentation, I would like to disclose here that some of the indicative workflows that I'm presenting are part of patent applications that we have recently filed. We have filed around 3 patent applications very lately. And our investigations and the projects and the extramural grants that we have been supported by Indian Council of Medical Research, Department of Health Research, Department of Atomic Energy, Department of Science and Technology, and Ministry of Human Resource and Development. So we have almost the kind of grants that a typical Indian institute or a setup enjoys too. So we have been heavily funded, and basically it's towards developing the nanobiosensors for various non-communicable diseases, including cancer. Now, in this slide, I'm going to provide you a highlight of the global burden of diseases. These studies that are being conducted by Institute of Health Metrics in 2021. If you look at the graph, it's quite clear that around 23% of global deaths, that's nearly 130 lakhs per year. 23% of global deaths, that's around 130 lakhs of the overall deaths that's seen in the world, can be attributed to environmental modifiable factors. Here we are talking of the particulate matter that is present in the air that we breathe every day, or the pesticides, remnant and residues that are present in the water that we drink every day, or any kind of environmental pollutants, heavy metals per se, that are infested with the food materials or beverages that we're taking every day. Combinedly, they have a huge impact on our health and disease index. So environmental modifiable factors are responsible for 1 in 4 deaths, almost at a global scale. And if we compare that with the Indian state level disease burden initiative, it is estimated by the same GBD studies, which is basically a 10-year-long study from 1999 to 2019. And as per the latest GBD estimates, it's around 18% of all deaths. So 18% of all deaths जो मौतें होती हैं हिंदुस्तान में, 18% only environmental pollutants or environmental pollution के कारण से हो रहे हैं. So this is just alarming, isn't it? 18% of all deaths that are taking place in our country can be attributable to environmental modifiable factors. So this forces me to, kind of introduce the word exposome before you. The combined effect of environmental toxic exposures, diet, and behavioral patterns combinedly can be taken together as exposure. It encompasses the totality of human environmental exposures from conception till death. A human being experiences from birth till death. This particular terminology was introduced by Dr. Christopher Wilde in 2005. 2005, Christopher Wilde introduced this terminology as chairman of International Association for Research in Cancer. He was the chairman of IARC, which is located at Blois in France. At 2014, Miller and Jones modified the definition to a bit and introduced the concept of biomarkers and signatures. So it now culminates into a zone in which we are not only talking of environmental factors, we are also encompassing all the dietary factors, all the kind of behavioral patterns. So the cumulative base
Mr. Sir, sorry to disturb you. Your slides are not moving. Hello. I think he's. Hello.
I think there is a problem in
There
internet.
is some technical issue with this, sir. No? He left the meeting. Yeah. Dennis, you just call to sir.
Yeah, I'm trying. I'm trying.
Please.
Hello. Yes, am I audible?
Yes, sir. Sir, one request was there. Your slide was not
Slide
moving.
is not moving, sir.
Oh, the slide is not moving?
So do not keep in the F5 mode. You just keep simply. Because
Okay.
sometimes when we keep in the full screen mode, they're not moving then.
Okay, so we were here till exposure?
Yes,
Yes,
sir.
sir.
Now
Okay,
it's
okay. So I also explained what exposure means, isn't it?
yes, sir.
Okay, so if we dissect the term exposure and look at the acute and chronic environmental stressors, be it short-term exposures like that happened due to the accidental release of methyl isocyanide gas at Bhopal, or the Fukushima radiation nuclear power plant accident, or the various kinds of acute exposures that we experience daily on daily basis. Chronic forms are the long-term exposures that present in the air that we breathe, or the water that we drink, or the soil and the food items that we eat every day. So together with nutrition and age-related factors, they kind of modulate our genome, epigenome, and metabolome. So, and they're responsible for the non-communicable disease sets, including reproductive ailments, which can be reproductive tract cancers. So this slide shows the temporal variation in exposure from childhood till death. So if the complete life scale is divided into 4 stages, can be divided into 4 stages, that is childhood, puberty, adult, and aging, the very impact of environmental toxic exposure begins as soon as the zygote is formed inside the womb. Those kind of exposures are termed as in utero exposure. Then we have accumulated exposure, occupational exposure, clinical exposure, and seasonal exposure. So all these 6 or 5 kinds of exposures, they together shape up the exposure of the human being. Here, since most of you are graduates and postgraduates in faculties of pharmaceutical sciences and nanotechnology, it is very important for me to say the fact that early life environmental exposures, whatever happens during the phase that we are inside the womb or during our childhood, has an immense impact on the adult disease state that we're experiencing. So early life environmental exposures can lead to later life health problems. There is not an iota of doubt that David Baker's hypothesis of developmental origin of health and adult diseases was conceptually right. So we are precisely what our grannies were exposed to. हमारे grandma जब वो young थी, जब उनको कुछ environmental exposures हुआ था, उसका भी फर्क हमारे ऊपर आज पड़ रहा है. So we are precisely what our grannies were exposed to, because these exposures can traverse in an epigenetic traits and using the transmitters of the biological phenomenon in a non-Jandalian manner. This slide shows the exposure response relations. So for any kind of environmental toxic exposure, be it the short-term or long-term exposure, they are going to have immediate, intermediate, and long-term impact. There is no doubt about it. Well, now it's time for us to debate and discuss whether biomarkers can offer really help. Can biomarkers really offer in predicting some disease subsets very early? So 3 things basically would matter. First is the time at which we are trying to take the samples or trying to study the individual. what kind of biological matrix that we're trying to study. Is it blood? Is it urine? Is it cerebrospinal fluid? And what is the best possible method that we are trying to, kind of understand the biological markers? So are we validating the biomarkers, or we are trying to develop some point of care tests? So when, what, and how are the three predominant factors, and that must be taken into account when we are discussing about biomarkers for early diagnosis of the disease. So for conducting any kind of molecular exposomic study, there has to be 5 levels: the kind of environmental exposures, the biomarkers of exposure, biomarkers of early effects, biomarkers of disease, and tissue bank. So, we can take help of the tissue bank in getting a lot of tissues and blood samples and urine samples. So the UK Biobank is one of the best examples. We have a cancer database biobank located at IIT Chennai and Tata Memorial Center, Mumbai as well. So these are the kind of ailments that one can see through, from the reproductive ailments point of view. It starts with the early pregnancy loss, spontaneous abortions, then single gene defects, chromosomal abnormalities, low birth weight, mortality, and various kinds of malignancies from the male reproductive to female reproductive parts. So I'm now going to give you a very small example of nanofine particulate matters that are present in the air, how they can really create a havoc by insinuating a process of irreversible epigenetic reprogramming. Can that really happen? Because these nanoparticles that are present in the air that we breathe every day, they contain a lot of polyaromated hydrocarbons that are emanated from the diesel exhaust. They contain a lot of heavy metals. So we were the first to demonstrate the kind of implication that these ultrafine particulate matter, or the nanoparticulate fine matter, would have on mitochondrial redox homeostasis. That means whether the cell's mitochondria is getting impacted by the constant exposure of ultrafine particulate matter, because ultrafine particulate matter can really be the pro-oxidants. They can help in generation of oxidative stress and a kind of sterile inflammation. So from there, we try to understand how this nanofine particulate matter that is present in the air can really go into our cellular system. So if you can just see here, so these particulate matters are coming into the phagosomes and getting fused with the lysosomes, and they create these free radicals, and the reactive oxygen species stress increases by the exposure of these ultrafine particulate matter. And no doubt they have genomic effects, mutations and other things that can really happen. But apart from the genomic effects, they can also have the epigenetic effects, which are basically regulated through mitochondria. So if genetics loads the gun, it's epigenetics which pulls the trigger. So this is a fantastic kind of representation. I always love to show it to my students and at all conferences that I happen to speak, because these are the laser scanning confocal microscope images in which the ultrafine particulate matter that is present in the air that we have been isolated and stained with acridine orange are being taken up by the cells here. You can see it, the red particles being taken up by the cells, and these are the nucleus of the cells. So this is the control 6 hours and 24 hours. So by around 24 hours, you can see the defined particulate matter is getting deposited inside the cells. So this is the live scale imaging, where particulate matter has been stained in acridine orange, and the mito tracker is being used to stain the mitochondrial components of the cell. So whereas you were basically looking at the picture and seeing yellow dots, these are the areas in which particulate matter is hitting and onto the mitochondria. They're going inside the mitochondria and trying to generate a kind of reactive oxygen species stress. So this is how the derailment of mito-epigenetic machinery completely takes place. I'm not going into the details because this contains a lot of complicated biological stuff in which many of you might not be interested. This is the autophagy detection by LC32 by laser scanning confocal microscopy, hypermethylation as observed by spectral karyotyping and multiphoton microscopy. This is P16 hypermethylation, which are basically the after effects of the particulate matter exposure. And this is a very beautiful slide again, in which the cells that were exposed, they went on to a phenomena of senescence. They did not divide for a longer time. And after that, suddenly, there were re-emergence of clones. These are all tissue culture-based experiments. So, see, the daughter cells were having properties of cancer stem cells. And these were basically injected into nude mice, the immunodeficient mice, which has a compromised immune system. So tumors started developing. So you can imagine the impact of this ultrafine nanoparticulate matter that is present in the air, how devastating it is. So these are again the aneuploid cells. And I'm basically, with this in mind, we thought that, epigenetics could be playing a paradigm shift role in reproductive health ailments, including malignancies of the reproductive tract, and whereas mitochondria must be playing an immensely important role, because inter-individual epigenetic variability, of course, it plays a role, but along with the genetic susceptibility of the individual, we can be predisposed to any amount of diseases. Because if you now see, the air pollution is being correlated with diseases of central nervous system as well, cardiovascular diseases, cancers, diabetics, and a lot of metabolic stuff. So how that is happening? In order to understand this, we started delineating the effect at a much molecular level. So we thought that it would be in an appropriate time to understand the ramifications at mitochondria, and the subsequent signaling patterns were studied by us. So here is the complete diagram of the workflow that basically was demonstrated, and we have published this in Current Pharmaceutical Design as a paper. Anyone interested can have a look at it. So this completes the first part of my presentation, in which it is extremely clear that mitochondrial mediated epigenetic modifications play an immensely important role in exposome analysis. And mitoepigenetic signatures may assist in potential shift from curative to predictive. हम इन्हीं markers का help ले सकते हैं. These circulating stuff or the circulating markers can really be extrapolated for developing the point of care test. So whether low cost innovations involving nanoanalytical framework can really be achievable, क्या हम बना सकते हैं point of care test? So that we are now going to discuss in subsequent slides. Now, what are nanobiosensors? Now, these are small nanoanalytical frameworks that comprise of nano-conjugated biological materials as transducing system. They have the ability or capability to recognize a minuscule quanta of biological, chemical, or physical analytes. They can detect these analytes at femtomolar concentration. So we were dealing with nano, then we came down to pico, now we have come down to femto levels. So these nanosensors can really recognize the analytes at femtomolar concentration as well. So what are the typical components of a nanobiosensor that needs to be understood? Because biometrics forms a fundamental part. Because आप क्या ले रहे हैं, whether it is urine, whether it is blood, whether it is cerebrospinal fluid, whether it is semen. So the biological metrics is going to play a fundamental role for all kinds of nanobiosensor studies. And then we have a transducer component and a detector component. Transducer often acts as an interface, measuring the physical change that occurs with the reaction with the bioreceptor. वो एक interface की तरह काम करता है, and detector basically receives the signals, amplifies, and subsequently the data is converted as digital signatures. which can then be captured by fluorometry, by colorimetry methods, by flow cytometry methods, by confocal microscopy, by any amount of methods. So basically, we have a transducer, we have a detector component. So nanoparticles play an inevitable role in all these fundamental nanobiosensors, because we are going to take help of semiconductors like quantum dots or any kind of nanoparticles that you can use based on the surface properties and the surface modifications that can be done. So nanoparticles with biorecognition elements attached to the surface specifically binds to the analyte, because after modifying the surfaces, it will recognize the bioanalyte. And then you can really get onto the transducer surface and amplify the signals that you're receiving from these analyte and the nanoparticle composites, and then analyze the whole results with the help of cutting-edge technologies like laser scanning, confocal microscopy, or flow cytometry. So I'm just going to highlight and provide you one specific example wherein we took help of fetal nucleic acids in maternal plasma. That means जो बच्चों के डीएनए होते हैं जो माँ के सर्कुलेशन में पाया जाता है. So post 14 weeks, we start getting the fetal DNA in maternal circulation. So around 10% of cell-free DNA in maternal plasma is of fetal origin. So we started realizing that अगर हम उस DNA को, if we can study those DNAs and the associated biological bioentities, then we can predict the kind of disease susceptibility that a child is predisposed to. So knowing well that the half-life of these molecules are less than 20 minutes. They can go undetectable to 2 hours postpartum, and there are ample number of studies in which fetal DNA is being used for RHD phenotyping. So we isolated the circulating nucleic acids of the fetus by using 2 commercial kits and by modifying the whole protocol. So first of all, we lysed, then we helped the circulating DNA to bind. After doing a centrifugation step, we eluted the DNA. So, and then we characterize the cell-free circulating nucleic acids that is present in the mothers. So they were around 500 base pairs in size, and we could also profile the histone that are associated in the nucleosomal core components. So histone 1, 2A, 2B, 3, and 4 were characterized. These are various kinds of microRNAs. MicroRNAs are 20 mer sequences which are there in circulation. So we plexed around 74 microRNAs from the fetal cells and then started looking at the indices by real-time PCR methods. We also looked at the mitochondrial DNA modifications and started characterizing the mitochondrial DNA, because mitochondria is the only organelle which you get from your mother. So that has a maternal lineage. तो आपके जो नानी थी, आपके मम्मी के पास आया, वो मम्मी से फिर basically the child is getting it. So it is extremely important to understand what kind of modifications, what kind of changes that these mitochondrial DNA have in order to understand future disease susceptibility. So these are the mitochondrial DNA fragment analysis that we did by genomic Sanger sequencing. And because the mitochondria DNA is around 15.8 kb or almost 16 kb in size, it's a circular DNA. And basically we started doing the genomic analysis by using the Sanger sequencing, and followed a 9 fragment analysis approach. So this is the mitochondrial DNA methylation studies that we conducted at our lab with 12S TF, 16S D loop and cytochrome B. Mitochondria के different regions को हमने फिर start किया analysis करना बच्चे का, this is the mitochondrial DNA of the fetal cells. Then we started developing a photonic dual nanohybrid assay for detecting the cell-free circulating mitochondrial DNA of the fetal cells. This was published in JBPA. So we isolated the cell-free circulating nucleic acids, as I just mentioned. Then we had a CQD-APA. There was a carbon quantum dot nanoanalytical framework, which was conjugated with aminopyrine. Aminopyrine has the inherent property to bind to all nucleic acids because it's an intercalating dye. So CQD-AP provides a blue-green fluorescence, and these aminopyrine molecules can be really conjugated, and the overall fluorescence that we get is aqua. So we started acquiring all these signals from the CQD-AP nanoanalytical frameworks by helping it to bind to the kinds of circulating nucleic acid stuff, all kinds of circulating nucleic acids. And we had a different nanoanalytical framework, which was of quantum dot, and which was labeled with the bioprobe. So the bioprobe was actually labeled with a fluorochrome, and these quantum dots were 705 nanometers in excitation emission spectra. So हमारे पास पूरे DNA को recognize करने के लिए there was a green fluorescence pattern, and for the mitochondrial DNA जो बच्चे से हमने isolate किया, that is, for that we had a red fluorescence spectrum. So green versus red. So fabrication of the capture and screen facet of the fluorescence-based dual assay took place, and wherein we allowed the pi staking to take place, and then carbon quantum dots were labeled with aminopyrine. We also studied the gel electrophoretic mobility assay, because if the conjugations are not taking place at the optimum level, then it is very difficult to ascertain that the fluorescence is coming really from the conjugated nanoanalytical frameworks and not from the background noise. So therefore, we conducted gel electrophoretic mobility assay for CQDs, 1 aminopyrine, CQD-AP, quantum dot 705, which is a red fluorescence, probe attached to 705, and then the total nanoanalytical framework. These are the co-localization studies studied by multiphoton microscopy, because in order to understand the structure of these nanoanalytical frameworks, which are almost around 20 nanometers in size, you must have a very powerful microscope at your place. So these are the blue clusters of CQDs. When we conjugate that to AP, we get an aqua green fluorescence, and then the dual colored clusters of CQD-AP and the captured by CQD-AP and propydium iodide. And these are the quantum nanocytometry systems that we developed with the help of a photoacoustic system of Thermo, and we tried to modify the flow cytometry complete unit for that can best suit for our purpose. So these are the development of integrated nanobiosensors. We have been very lucky to have collaborations with Saratov State University Russian Federation, Neufer Institute of Occupational Medicine Poland, Ghent University Belgium, and Northwest University South Africa. So the test that we have developed for looking at the mitochondrial DNA deformities of the child when he or she is already in the womb. So we have named that as epimitomipt, which is a 3R concept, because it has reduced risk, because almost it's non-invasive. We are not trying to harm the mother in any means. It has a very reduced cost and reduced maneuvers, because we only need to have multiphoton microscopes and nanoacoustic systems, best flow cytometry systems to understand the fluorescence lineage and the fluorescence patterns. There are other kinds of nanobiosensors that we are involved in. There's a development of the search-based nanobiosensors, oligonucleotide biochips. Also, we have started working on it. And so the message of part 2 of my presentation could be a dual nanophotonic assay as a single unit miniature platform offers robust, reliable, and sensitive identification of candidate biomolecular signatures like fetal DNA that is present in the metabolic circulation. An easy to use biosensing platform capable of precisely detecting 2 molecular signatures with utmost specificity, sensitivity, and reproducibility, because CQD-AP was detecting whole nucleic acids, and the QD705 with a bioprobe was detecting the mitochondrial DNA component. So, but the question remains, with the same degree of accuracy can be obtained using biodegradable, environmental friendly semiconductor nanocrystals, because we are an environmental health institute. So we need to develop nanoanalytical frameworks by following the green protocol, green chemistry based protocols that we are currently doing. So our lab has been able to demonstrate the fundamental principles and the applications in almost all leading journals that is there in the Atlanta, trends in analytical chemistry, biosensors and bioelectronics, luminescent quantum dots, mRNA detection again in Atlanta, nanomedicine. So we have been able to capture cell-free circulating DNA, cell-free circulating microRNAs, cell-free circulating mitochondrial DNA, and cell-free circulating LNC RNA. Once again, list not at the last, but I would like to thank Dr. Dinesh. He's a long-standing collaborator of ours, in which basically me and Dr. Dinesh have worked very closely for developing dendritic cell-based nano-engineered cells. When I was there at Kumar Memorial Hospital as well as in Tata Memorial Hospital, we had a lot of funding, and Professor Nk Genser was also associated with the collaborative effort. So I thank you, Dr. Dinesh, Dr. Pankaj, Arun Bhatnagarji, and Dr. Subit Kumar for letting me speak in such a nice manner, giving me the opportunity. Thank you, Chairman. Thank you.
Thank you.
Thank you.
So thank you, Mishra sir, for giving such a wonderful lecture. And what I feel that your first bird of your topic, that is a development of the low cost innovation. That is a very, very important one, especially if we want to utilize this nano formulation, nanotechnology for humankind. Once we are able to reduce this cost and these types of the technology as you are developing, not only in the particular area, but it is serving for whole humanity. Means some things, if we can able to detect the disease earlier, that was your topic, it is going to be the great help for the whole community and the whole world.
I agree.
So
I agree.
this types of the research and innovations are very much required. Doing in the particular area, probably sometimes we are getting narrowed down. But scientists like you, when you are thinking for not only for particular area, but for the whole India and for whole world, and the concepts of the early detections of the any of the disease is very, very important. But we know it during this COVID, how much means loss we are facing, because we have not prepared for these types of the disease. So now ICMR and DBT, everybody are now ready. Now they are funding for this ABCL facilities, BSL facilities. So now these types of the some new horizon are coming. Probably now we are preparing well for our future. And once we having a very good preparations, probably this much loss of the humanity is not going to be done. So thank you very much, sir, on the behalf of the IIPA, on the behalf of the Dinesh Mishra and his team, and Rekha also. I'm highly thankful for you to sparing your valuable time and to give us such a nice speech. Thank you very much, sir. Nice time. Thank you very much. Thank you.
Thank you very much, sir. Now, I would like to request our respected principal, Dr. Dinesh Kumar Mishra, sir, to felicitate speaker of scientific session and express token of gratitude to the chairperson, Subit Kumar Genser. Thank you, sir. So over to you.
Thank you, Dr. Rekha. So it's always pleasure listening Dr. P.K. Mishra, sir, as I have got an opportunity to work under his supervision. And my senior is also there, Dr. Subit Jain. He guided me well during my post-graduation and during my PhD work. So I'm very thankful to both the chairperson and speaker. And on behalf of entire IIP and organizing team, please accept our token of appreciation and token of love. Dr. Mishra, please accept. Thank you very much, sir. And Dr. Subit Jain, sir, please accept our certificate of appreciation. Thank you very much.
Thank you
For sparing your valuable time. Thank you very much, sir.
Thank you. Thank you, Mr. Ji, for inviting me. It's my pleasure. Thank you.
Very much, sir. Now, moving ahead, I would like to welcome again Dr. Pradeem Kumar Mishra, sir, as a chairperson of the last scientific session of today's.
Dr. Rekha, please, sorry to interrupt, but I think I'm trying to connect Nk Genser. Some technical issues there. Just wait for 1 more minute.
Sure, sir, sure.
Yes. Nilesh ji?
Yes, sir.
Can you check? Professor Nk Jain is trying to connect.
हाँ सर अभी तक नहीं आया वही
Dear participants, Dr. Nk Jain sir is joining us soon. So meanwhile, I just request every participant for the oral session, kindly be prepared with be ready with your PPT. As your turn will come, you will be promoted as co-host and you'll be able to present your PPT. So I request everybody to get ready with your PPT. And after this session, we'll be having lunch break of 30 minutes. And then after lunch break, we'll start with the oral session. Thank you.
Dr. Rekha?
Yes, sir. Yes, sir.
Yes, sir has joined.
Okay, fine. So, continuing with the scientific session of day one, I would request Dr. Pradeem Kumar Mishra, sir, to please introduce our eminent speaker, Dr. Nk Genser. Sir, please, over to you.
Sir, namaskar. It's
Namaskar.
it's my proud privilege to introduce Professor
Yeah,
Nk Genser.
I'm very happy.
Professor Nk Genser is a very learned professor and a professor of distinction. Professor Jain actually started his academic career in 1973 as a lecturer in pharmacy, Hulk College, Indore. Subsequently, he served as reader in pharmaceutical technology at MS University, Baroda. And subsequently, he was at Nasik and the head of the department of Dr. H.S. Gore University. And basically, we also collaborated with Sir and Dr. Dinesh during his tenure at the Department of Pharmaceutical Sciences, Dr. H.S. Gore Central University. We were quite lucky to have his blessings and his guidance for our dendritics and engineering. Thank you so much. Professor Jain is the author of 24 celebrated books in pharmaceutical sciences. So,
This one is to remove.
a prolific series on controlled and novel drug delivery systems, and has many publications in
Connecting.
Yes, sir.
So, Sir has contributed more than 50 chapters to national and international books. He has to credit 480 number of publications. 480. Sir, you are a living, true legend in your own standing. So, in reputed pharmaceutical journals, and supervised 54 PhD students and 142 M.Pharma candidates. His total citations are 54,843, and his publication rate index is 111. Professor Jain has been the recipient of IGMA Research Award in Industrial Pharmacy,
The same thing.
Devi Dandia Award, Prize, Best Paper in Pharmaceutical Biopharmaceutics published in IJPS, Memorial Award, and several such awards to his credit. Professor Jain has been associated with professional associations, particularly AICTE, IHPA, IPA, IPGA, and served in many distinct committees in UGC, AICTE, PCI, NAC, NBA, and the most of Indian universities, and is an expert. Sir is considered a true legend in the field of pharmaceutics, nanopharmaceutics, and pharmaceutical biotechnology as well. He is acclaimed worldwide for his significant contribution to pharmaceutical research, teaching, and administration. He's extremely popular within the student community of across states. At various conferences, he speaks very eloquently for hours together. I have been lucky to have heard Professor Jain a number of times at various podiums. So, with these words, sir, I would like to thank you for accepting the honor of Dr. Dinesh and providing me an opportunity to introduce you. So I feel extremely blessed. Thank you, sir. Thank you.
Thank you so much. Thank you.
Over to you, sir. Over to you. The floor is yours.
Hello.
Yes, sir. Audible, sir.
I am audible. Okay. I'm still struggling with my presentation, but then I'll start. Okay.
Sir.
Yeah. So thank you very much. Thanks, Professor Mishra, for nice introduction. I am also thankful to organizers for inviting me to join this. And at the same time, I am happy that this program Hello. Hello.
Sir, that echo sound is coming. Sorry for the interruption.
Hello.
Yes, sir.
Stop. Hello.
Yes, sir. It is better now, sir.
Hello.
Yes, sir.
Screen sharing.
Yes, sir. It is coming, sir. Your voice is also coming. The screen is visible.
Hello.
Sir.
Hello. Hello. Screen sharing is there. Yes. Hello.
Sir, you are audible, sir.
Yeah, thank you very much. Thank you so much. Yes, how will it go? Okay. So the topic which has been selected, that is really relevant to the pharmaceutical research today. The topic basically, it is related to the research. It is also related to the translational research, and it is also related to the nanomedicine. So before I start, I should try to tell you that translational, that is related to translation. Translation, we are all familiar, and it is related to the translation of the information, particularly the scientific information, into the practical applications. This is also, so that is the translational. And now coming to translational research, this is used interchangeably with the translational medicine or translational science, or to be more popular, to make it more understandable, that is actually translating in pharmaceutically from bench to bedside. So this is an effort built in basic scientific research to create new therapies, medical procedures, or diagnostics. So the word translation is used here very specifically to talk with the medical, say, terminologies, and that's why new therapies, medical procedures, are diagnostics. This is what depicts the translational research. So we start from the basic research and we reach to the clinical research. This is the real meaning of the translation research so far as the pharmaceutical applications are concerned. So this is taking it from bench to bedside, and what is involved in the basic research, what is called the omics. These are the examples of genomics, proteomics, and metabolics. And ultimately, we have to enter into the clinical research. There is the clinical trials, epidemiology, and many, many other things. So this is way how we translate right from basic research to applied research. And in pharmaceutical terms, new products and approaches, what are the gaps? These are to be identified first. And then in implementation, what are the gaps? These are to be identified. So here, what I'm showing, This is the basic infrastructure so far as the translational research is concerned. So we start from the clinical basic research, clinical, preclinical, and then clinical. Finally, we go to the clinical implementation, and this is what is required for the public health in current times. To be very precise, the phases of translational research, they are divided into the 4 terms, the T1, T2, T3, T4, and these are the descriptions and the examples. For example, in case of T2, this is concerned with the phase 3 clinical trials. I am very sure that you people are familiar with the phase 1, 2, 3, and 4 clinical trials, because this is a requirement today for any pharmacist to understand. Those who are the teachers, they must be knowing. Those who are the students, they will try to learn gradually, because now this is to be a part of your B.Pharm curriculum, and hence it is very important for all the pharmacists to be familiar with this. Translational research to drug delivery systems, this is in fact aggravated with the help of what is called as the PD, or the product development. And this product development, this is we have to correlate with the nanomedicine. The topic which I am discussing is the translational research in nanomedicine, and therefore I would like to take a couple of minutes to introduce you to the nanomedicine. So it is the application of nanotechnology for medical purposes. That's why it is nano and medicine. It is use of nano materials for diagnosis, monitoring, control, prevention, and treatment of diseases. These various definitions are there so far as the nanomedicine is concerned. But I would like to add to the last one, that is the nanomedicines are medical applications. Meanwhile, it is medical applications of nanotechnology in treatment, diagnosis, prognosis, and control of biological system. That's why I said that this topic is very relevant. It's a contemporary topic so far as the pharmaceutical applications of nanotechnology, nanomaterial, nanomedicines are concerned. These are the aspects of the nanomedicine, but try to remember that whole effort of developing the nanomedicine is going to lead us to realize what is known as the concept in drug delivery, that is the magic bullet. Magic bullet, this is the concept was given by the Paul Ehrlich. And the basic idea is that the drug should be designed in such a manner that they can be administered and they reach only the target and nothing else except the target which is required for the drug to reach, so that all the requirement of drug delivery is satisfied in terms of overcoming the non-desirable effects, side effects, etc. So the drug reaches only at the desired site or the target, and that is just like a bullet, and it is a magic bullet because it is reaching only at the specific site. That is the realization of the concept which was given by the Paul Ehrlich. And those who are interested in having, latest drug discovery and the latest areas, they will assure that the concept of magic bullet is also realized. And that is in one way basic objective of having the translational research. So we want to make the translational research, particularly in case of nanomedicine, so that we have all the possible benefits of a drug without and devoid of any undesirable effects. In your high school or higher secondary school, you have come across these terms. I am just trying to remind you that this is the nano. This is concerned with 10⁻⁹ meters, and this is not the limit because we are likely to go to pico and femto, although the size is going to decrease. Yet the quality and the quantity of the material that is going to change drastically if you understand what is the meaning of the size, or what is the effect of the size, or what is the effect of size on drug delivery of a particular material. So in case of clinical development of nanomedicines, there are certain key issues. And these key issues, I'll request all the participants to understand very carefully. And now here what we include, there is the biological challenges, large scale manufacturing, biocompatibility and safety, intellectual property rights, regulations, and overall cost effectiveness. Whatever you do with your drug, and if it is not cost effective, you can understand that it will not be marketable, or it will remain a sort of a drug which is not acceptable because of the cost itself. So these factors, they impose significant hurdles, and we will try to overcome them so that the drugs are really useful. Whatever translation we have done, the translation can be implemented. So for implementation purpose, one of the criteria is the cost. So what are the challenges to the nanomedicine regulation? These are some of the points which I have listed down. Lack of unified definition or classification of nanomedicines or the nanomaterials. This is a serious problem, because unless we are very sure what we would like to characterize, how we would like to characterize, it is difficult to proceed with the development of the nanomaterials. Then regulations across the globe by different agencies, they are also not uniform. Analytical methods, they differ from each one material, nanomaterial to each other. And the PQ profiles, they are also divergent. Stability issues are there. So like that, there are n number of problems with the nanomedicines. So it is very easy to say that we are talking about nanomedicine, but we do not really understand what type of problems we are going to face when we try to develop a nanomedicine, or we try to translate or translational research leading to nanomedicine. So the essential concept remains there, that when we talk about the nanomedicine, we are talking from taking a drug from bench to bed or the clinic. So this is basically concerned with the drug delivery, diagnosis, theranostics. Theranostic, that is the therapeutic and the diagnostic. So the combined word, which is very popular these days. So for the pharmacist also, it is easier to understand that if we have the two concepts, that is the therapeutic and diagnostic, both aspects are combined in one device, then it will be more useful. Then the further development of this field, they will face significant challenge in the regulatory level. Because once we try to translate some drug into a nanomedicine, we would like to market it, and before marketing, we have to go through the regulatory approval. And before regulatory approval, we should know what are the requirements of the regulatory approval, or the regulatory criteria for the approval, and then only we can proceed further. This process of the product development, which I told you, which comes in between say basic science and the product development, this requires a set of activities required to bring a new concept to a state of market readiness, and it requires a basic design. It is basically concerned with bringing something to the clinical application, and as pharmacists, we also know that the chemical entity or the medicament must have a quality, built-in quality has to be there. Now, if we can understand that we want to translate an idea of a new proposition to successful product for routine manufacturing and commercial marketing, in that case, we have to realize that the product development will consist of different aspects, which I am not going into the details because of the lack of time. But at the same time, this is also influenced by the development of the batch, which is really we understand as the pilot plant at the scale up. And this R&D to production with the help of pilot plant, this is the basic say, art in bringing about a real translational research. So the scale up, this is the art of designing a prototype using data obtained from pilot plant model. So for the clinical success, the larger scale manufacture will be required, and large scale manufacturing will require the scale up, and hence the total aspects of scale up are required. So in a nutshell, I can say that objective of pilot plant at scale up are say summarized in a simple way. What is that? Find mistakes on a small scale and make profit on large scale. So before going for large scale production, if you can identify the lacunae or certain drawbacks in the production of the product, then it will be the job of the pilot plant and scale up, or pilot plant and scale up helps us in bringing about this realization. Or another way it is mentioned as commit blunder on a small scale and make profit on large scale. Hope you will understand what I'm talking about. That before going for the large scale, we try to identify the lacunae at a small scale so that we don't commit the blunders. So these are the basic objectives of the pilot plant and scale up. I will not go into the details of this, but this is the importance of the pilot plant, and these are the functions of the pilot plant. To come to a better understanding of the translational research in nanomedicine, we have to consider some other aspects which are we may not be really familiar with that, but then we must try to understand. These are the concepts of the quality by design and the process analytical techniques. And the main objective is to design and develop formulation and manufacturing process, ensuring predefined quality. Underline this, predefined quality. So we have already defined the quality which we would like to achieve in a product. That is also a requirement for successful translational research. Now, this QBD or the quality by design, this is also another concept, and this is also used today, although we may not be understanding in academic circles yet. QBD is a method by which we can try to minimize the errors, and it is a concept for abbreviated new drug application that was developed by Zuron. QBD perspective means what? What really it means? Quality cannot be tested into products. It has to be built in by design. So at the end of production, if you carry out quality testing, that is not the proper way. That is not the scientific way. First, you decide what quality attributes you want in your design, in your product. Then you design the product in that formulation and try to incorporate all those attributes before you reach to the final product. So final product will definitely have the designed quality attributes, and that is the basic objective of a QBD, quality by design. Interpretation of quality, they differs from person to person, but while talking about the quality, we have to consider many aspects. Most of you are familiar, so I will not go into detail of this. Target quality profiling. What is the target so far as the quality profiling is concerned? You can see many things I have shown here. Try to understand it, and let's go to the next one. So what we really want quality to critical to quality are the critical quality concepts. These are this I have tried to show. This is what a user wanted, and this is what viewed by many others, and what the user actually wanted and how it actually works. These are the concepts, these are the precepts, and these are the ways by which different things are viewed by different persons, or the scientific say scientists or others. So another concept is the design of experiments, that is the DOE, and this is again meant to have a perfect product, and which is free from any defect, and the defects, if any, they have been overcome by proper design of the experiments. So experiment cannot be say unscientific. If we have designed something, we will proceed further, check every stage, and then proceed to get the final product. All this takes me to understand what really is basis of the GMP, GLP, etc. And if you can trace out the history, you can find that all this thing is started with the thalidomide. His story is very long, but then it is a very important for the farms to realize that this was a sort of scandal, thalidomide scandal, in which the children were born with say deformed say bodies, or deformed children were born because the mothers or the pregnant ladies, they had used this thalidomide, and it was not properly documented, it was not properly tested, but it was claimed that it could be used by pregnant ladies to overcome the morning sickness. But it was used in almost all European countries except USA. And in USA, it was not possible because the person, lady who had just entered the FDA, she said that you have to prove that it is safe in the offspring. It is safe for the pregnant ladies also, which they could not do it. And the company, Chemi, they tried to pressurize that lady, but she is not going to put her foot down. And that's how the whole country, US, was saved from the use of thalidomide, and the deformed babies were not born in US. But that was there is it will be a great achievement. Then certain concepts are there which I think in translational research in nanomedicine will have to consider. As I told you, regulatory considerations are one of them. The GMP, GLP, that is good laboratory practices, GCP, good clinical practices. These are the newer concepts you have to understand. All the statistical concepts we have to understand, how the documentation should be done, how the validation should be done, and how the optimization should be taken. Another area which is very much highly rewarding, that is the artificial intelligence, and it's not something new. We have been using this term in terms of the calculator. We have been using in terms of the computer games. So we are familiar, but yes, it is not being practiced to that extent in translational research. So in translational research, artificial intelligence are very useful, and those who have some idea about the artificial intelligence, you can understand that it will save a lot of time in bringing a drug from bench to bed, or the overall time required for development of a drug product, bringing it to the market, that will save to a very large extent if we apply the concept of artificial intelligence. Simultaneously, we should also use quality measurement system, that is the QMS, for next generation nanomedicines. And this is again going to be a scientific basis for the development of the translational medicine. Artificial intelligence, I'm not going to into the details. But AI is not only transforming the way the pharmaceutical industry is evolving, it is also offering drug manufacturing companies innovative ways to enhance the brand value. That is the real application of the artificial intelligence in pharmacy. This I am suggesting to all of you, although most of you have gone through, have heard about it, but then new drug and clinical trial rules 2019. This is effective from 19 March 2019. And this is superseded Part 10A and Schedule Y of the drugs and cosmetic rules with immediate effect. Now, these rules apply to new drugs, investigational new drugs for human use, clinical trial, bioequivalence study, bioavailability study, and ethics committee. But why I'm telling you, because now this is to be a guideline for development of the new drugs. And if it is a guideline for new drug, naturally it is useful for the translational medicine, translational research leading to the development of the nanomedicines. And hence you cannot avoid the clinical trials because whatever you are developing now, that will be new drug, and hence for them clinical trial rules are to be followed. And that's why for translational research in nanomedicine, all these things are to be looked into. So with this, I think I would like to conclude, because I got less time. I am also trying to be very fast. But I must say something that in case of translation research, we have to be careful and try to understand what we are trying to develop and how are you going to develop. This is something which is a bigger challenge, because I have a question to all of you. Try to understand how many PhD theses are produced, how many MPharm theses are produced, how much efforts are put in the research, carrying out this research, etc. How many publications are there, how many patents are there, and what is the real output. That means whether the work done in the laboratory is translated or not? And if it is not translated, then is it a futile exercise? Or what is the real outcome of the research being carried out in the academic institutions, particularly MPhil and PhD level, that everybody should ask. And that leads me to a suggestion that whatever we do, we must know what is expected outcome of that. What are we going to contribute? Are we going to contribute anything in terms of the development of healthcare system, or is it just an academic exercise? This is all the question we have to ask ourselves, and we have to convince ourselves that we are not wasting the time and we are not wasting the taxpayer money in terms of the scholarship which we are getting on the funding agencies, etc. So once again, I would like to thank you for the participating and the organizers for giving me this opportunity to introduce you very briefly to a topic which is very, very important in the current context and which will be important in future as well. So I would say appreciation to the organizers, and thanks to all the persons, including Professor Dinesh Mishra and Professor Pradeem Mishra for my introduction. Thank you very much. All the best. Thank you very much.
Thank you, sir. Thank you, the organizers. I thank the organizers for providing me again a wonderful opportunity to listen to a true legend of pharmaceutics and nanopharmaceutics. What I could gather from Professor Jain's slides and the presentation broadly can be categorized into three aspects. The first message could be translation of research into clinical practice can be conceptualized as a proceeding from awareness through acceptance to adoption. So, sir wanted to highlight these aspects in a very different format, but translation should also be considered in research design. That what sir emphasized a multiple number of times, including the end users and an evaluation of the research implementation. And he categorically also mentioned that quality cannot be tested into the products. It has to be built in the design. So you have a faulty design, the product is also going to be of no use. So he also suggested all of us to be cautious in using the word translational research overtly and outwardly, and conduct research in a very authentic and in a right spirit from the real manner in which it can go from lab to practice. Thank you, sir, and thank you, organizers, for providing this opportunity. It was wonderful. Thank you.
Dr. Rekha?
Thank you, sir. Thank you very much. Now, I would request our respected principal, sir, to felicitate Dr. Nk Genser and express token of gratitude to the chairperson, Dr. Pradeem Kumar Mishra, sir.
Thank you, Dr. Rekha. And I'm fortunate enough to, being a student of Professor Nk Genser, to felicitate on this occasion. I'm really thankful. It's my sincere thanks, because, just to tell you that sir he's traveling and he's a committed teacher. He's a true academician while he was traveling, but since he committed that he will deliver a talk. So in spite of so many troubles, he gave a very nice lecture. So I'm very thankful to Nk Genser. Thank you very much. And please accept our certificate of felicitation. And thank you very much, Dr. P.K. Mishra, sir, for sparing your valuable time, because you're also too busy. So thank you very much, sir. And please accept our certificate of appreciation. Thank you, sir.
Thank you, sir. Thank you very much. Thank you all the dignitaries for giving your precious time for this conference and giving such a wonderful informative sessions. Thank you all. Now, all the participants are requested to join the session for oral presentation at 2:30 p.m. sharp, and kindly be ready with your presentation. Thank you very much. We'll be having a lunch break. Thank you.
Recording stopped.